Yeah, thank you so much, Vivian. So my name's Breeya, was introduced earlier. I recently graduated with my master's in bioengineering, and I'm currently a technician in Dr. Rudy Tansu's lab at Mass Gen Hospital. And so I'm going to introduce our second speaker, Ilana Gabanyi. And Ilana did her PhD research at Rockefeller University. She's currently a fellow at Pasteur Institute, and today she'll be speaking on the crosstalk between, of course, gut bacteria and neurons. So please, Ilana, take it away.
Thank you. Let's see. This is gonna work well. Can you see well?
Yes.
Can you see the mouse arrow? Okay. So hi everyone, thanks for connecting, and thanks the organizer for inviting me. I think this is a great opportunity and I'm happy to contribute somehow. Since my PhD, I'm very intrigued about the crosstalk between gut bacteria and neurons. And what I also find amazing about science is that it's actually endless. Every work, every discovery paved the way for new and more complex questions. In 1683, the Dutch microscopist Antony Leeuwenhoek decided to take a look under his very impressive handmade microscope of a sample of what he found in between his teeth. And quite surprising for the time, he found living creatures in this sample, what he called animalcules. By studying his drawings and the description of these animalcules, we know nowadays that he was actually describing bacteria for the first time. 200 years later, the Italian scientist Camillo Golgi developed the silver staining method and described and drew for the first time the human neurons. And here we are, 150 years later, after the many questions that these two discoveries led to, trying to understand how these two very distinct types of cells are able to communicate. And to study this bacteria-neuron interaction, I decided to dig deeper into the mechanisms of the gut-brain axis. In other words, how the bacteria present in our intestine is able to influence the brain and what are their downstream consequences. Many studies have implicated the microbiota and their effects on different brain-related disorders. Most of these studies, as Brittany mentioned, are unfortunately still only correlation. For example, if you give the microbiota from an obese mouse to a lean mouse, this mouse would gain more weight. Also, psychiatric patients usually have some problems in their microbiota. But the possible therapeutic implications, if the mechanisms are reviewed, are very encouraging. And Brittany just showed us a very nice example of that. During our lifetime, our microbiota goes through important changes, both in number and in diversity. It increases until adulthood, and then it starts to decline again. And this process is concomitant with distinct neuronal processes occurring during specific stages of our life, pointing to an intimate relationship between gut microbiota and brain homeostasis. What is interesting is that our gut also changes our gut microbiota also changes in a much narrower time scale. It is actually under constant change, following the feeding cycle and the circadian cycle, where not only the number of bacteria changes, but also the type and the metabolites they produce. These metabolites are able to reach the circulation and affect distant organs, such as the brain. Most of the mechanisms describing how the gut microbiota and their metabolites are able to affect the brain involves a third party. Bacterial compounds such as short-chain fatty acids, neuroactive compounds, or structural compounds of the bacteria such as Peptidoglycan or LPS are described to influence brain-related behaviors either via the vagus nerve, interactions with the immune system, or with the gut epithelium. The gut epithelium, in response to these gut-derived metabolites, are able to produce hormones that in turn reach the brain. What I'm going to show you today, and the novelty my work brings into the field, is the description of a direct bacterial compound brain neuron interaction, where peptidoglycan fragments released by the gut bacteria are able to reach the brain and alter neuronal fiber. But before I go into the details of my work, let me introduce you the peptidoglycan and tell you why I thought that this was a great molecule to be a candidate to be a gut-brain signaling molecule. First, it is highly abundant. It is present in every bacterial cell wall, both gram-positive and gram-negative. Second, it is regularly released at the steady state. The bacteria, once it has to grow, to divide, or once it dies, it has to break up its peptidoglycan layer, and this peptidoglycan is recycled in a process called PG, peptidoglycan turnover. But this process is not 100% efficient. Therefore, there is the release of these peptidoglycan fragments, known as neuropeptides, into the environment. Third, it was already known that peptidoglycan worked as a signaling molecule among bacteria themselves. And since peptidoglycan is such an important and abundant bacterial component, different ways of detecting it was generated during evolution. A more primitive detection system are the PGRPs, peptidoglycan recognition proteins. These proteins are able to bind to the bacteria in linking to its peptidoglycan layer, and they are able to induce cell lysis, therefore killing the bacteria. Another more complex system present only in vertebrates are the node-like receptors. These receptors were first described in immune cells and are mostly known for its pro-inflammatory response. Node 1 responds to fragments that are present only in gram-negative bacteria, while Node 2 is a more general receptor because it responds to fragments present in gram-negative and gram-positive bacteria, such as the muramyl dipeptide, MDP. Although mostly known to induce this pro-inflammatory response during bacterial infection, more recent work have highlighted that not only peptidoglycan fragments are released into the circulation by the gut microbiota under homeostatic conditions, but also that these fragments play an important role in cell development and homeostasis. For example, it affects systemic T cell development. In the bone marrow, it is able to affect neutrophil development, and moreover, it affects neuronal development. Regarding more specifically the peptidoglycan effect on neuronal activation, the studies in Drosophila indicated that the peptidoglycan could have a direct effect in neurons, what was very interesting to me. In vitro studies showed that upon inducing a synaptic release with PHTX, neurons from flies that didn't have the PGRP did not recover as well as neurons from a wild-type fly, indicating the role of peptidoglycan in presynaptic homeostasis. Another beautiful work demonstrated that under a scenario of bacterial infection, the peptidoglycan would somehow decrease neuronal activity, and this decrease in neuronal activity will lead to a decrease in egg laying, most likely to save energy to fight the infection and to avoid the spreading the infection to the newer generation. So in order to reveal if peptidoglycan fragments known as neuropeptides could indeed work as a gut-brain signaling molecule under physiological conditions and directly affect brain neurons, my project needed to answer three main questions. Do neuropeptides reach the brain? How and if they alter neuronal activity? And what are the downstream effects on metabolism and behavior? So how did we answer those questions and uncover this new microbiota brain pathway where bacterial sensing via neuronal node 2 is able to regulate appetite and body temperature? First, I showed that the neuropeptides that are part of the peptidoglycan present in every bacterial cell wall were able to reach the circulation and the brain. And in order to do so, I colonized mice with radio-labeled bacteria. This labeling was exclusively on the neuropeptides. And 24 hours after colonizing these mice with this radio-labeled bacteria, we analyzed the brain and their blood. And as you can see in this graph, we detected both in the blood and in the brain radioactivity, implying that indeed the neuropeptide coming from the gut was able to reach the circulation and the brain. Interestingly, we saw that in females, there was a higher accumulation of neuropeptides in the brain. I also demonstrated using the hchiu GFP mouse that the hchiu receptor was present throughout the brain in several brain regions, including the hypothalamus. By co-labeling GFP with IBA1, a marker for microglia, we could show, as expected, that microglia would express the hchiu receptor throughout the brain. Surprisingly, at the time, we saw by co-labeling with neun, a marker for neurons, that those neurons also express the hchiu receptor, however, not evenly throughout the brain. We did detect it in the striatum, in the thalamus, in the hypothalamus. However, for example, in the cortex, we did not detect any neuron that expressed the hchiu receptor. And interestingly, we did not observe any neuronal hchiu expression in the gut. In the intestine, most of the hchiu expression was linked to the endothelial cells, here labeled with CD31. No colocalization was seen with TUS1 that labels the enteric neurons. So this neuronal hchiu expression in the brain was not restricted to a specific area or to a specific neuronal population, since hchiu neurons of different sizes and from different areas were found in the brain. So in order to study the role of this receptor on brain neurons, I decided to take a broad approach. And by using the Krylov system, I generated two types of mice. One in which the hchiu expression was removed from the inhibitory neurons, the VGAT hchiu mice, and another one in which hchiu expression was removed from the excitatory neurons, the CAMK mice. And by using these newly generated transgenic mice, I observed that in the absence of hchiu, females starting around 6 months of age would gain more weight than their controls, and no phenotype was seen in males. In order to know if this weight gain was related to appetite, we analyzed also how much these mice would eat. And we saw that indeed older females would eat more than their controls. However, we didn't see any appetite difference in younger females, neither in males. And to confirm the role of neuropeptides in this increase in appetite, we gave neuropeptides to the mice orally and observed that control mice, after receiving this neuropeptide, would eat less, while the conditioned knockout mice would eat more, showing that indeed hchiu and its ligand NMT are implicated in feeding control, and that most likely neuropeptides work as a satiety signal only in female mice. I also observed that these mice build nests less often than their controls. And to quantify that, I would give the mice a cotton nestlet, and a few hours I would analyze how much of this cotton they had unrolled to make the nests. And as you can see from these pictures and from these graphs, the control mice would build nests often, while the VGAT hchiu mice wouldn't do that as often. And as you can see here, the difference was greater in older females, although already detected in younger females. We know that in mice, nests are also used for temperature control. So we went on and analyzed using subcutaneous probes the body temperature of these mice. And we could see that the maximum temperature they reach over the day was the same. However, we did detect a difference in the minimum temperature they reached, meaning that actually these mice were warmer, so probably they didn't need to build the nest to increase their temperature. And given this difference in their minimum, they would have a lower variation in their body temperature. And no difference was found in males, nor it was found in the CAMK hchiu mice. We didn't see any difference in weight gain, food consumption, or in nest building. revealing that not-Chiu expression on excitatory neurons is not necessary for appetite or temperature control, while its expression in inhibitory neurons is important. So we have shown that gut-derived neuropeptides are able to reach the brain. Brain neurons express its receptor, the not-Chiu receptor. And if you remove this not-Chiu receptor from the inhibitory neurons, you develop certain physiological consequences. And that finally, that MDP would induce satiety. But none of this explained whether MDP would affect or not the neuronal signal. So in order to answer this question, we went on and used the iDisco clear map system. This system allows you to analyze the whole brain in an unbiased way. You clear the brain and you stain for the FOS marker. The FOS is a proxy for neuronal activation. Then, using light sheet and the clear map pipeline, you are able to analyze the brain and obtain the images of where there is more or less neuronal activation. Also, you get an Excel table that you can turn into volcano plots with the number of neurons in every brain nuclei. So you can visualize here the nuclei that were significantly affected when you compare mice gavaged either with MDP that affects the not-Chiu neuron, or the MDP control that does not activate the neurons. And with this, we could show that indeed MDP would affect neuronal activation. But moreover, we showed that this activation was sex and age dependent. Regarding age, you can see that in younger females, MDP had barely any effect, while in older females, it affected many brain nuclei. However, these affected brain nuclei were not the same as the affected ones in the males. Since our phenotype was in older females and it was related to food consumption, we got interested in the arcuate nucleus of the hypothalamus. This nucleus is known as the main controller of food consumption. And also, more interesting to us, it also expressed the not-Chiu receptor on the inhibitory neurons, here labeled in purple. And by using different techniques, I reviewed that the activation of the not-Chiu receptor by the neuropeptide MDP induced a decrease in neuronal firing in the arc. First, as I shown in my previous slide, by using iDisco clear map system, I reviewed that the number of FOS positive neurons in the arc was reduced upon MDP gavage. However, this technique doesn't tell you anything about which type of neurons are being affected. Therefore, I decided to use fiber photometry, in which by injecting the fluorescent indicator, calcium indicator, GCAMP, exclusively on inhibitory neurons of the arcuate nucleus, we could analyze in vivo in freely moving mice the activity exclusively of inhibitory neurons of the arcuate nucleus. It's known that these neurons are highly active when the mouse is hungry and are quickly decrease their activity upon food presentation. So in order to see if we had injected in the right place and implanted the fiber in the good place as well, we first analyzed whether we could reproduce this phenotype. And this is what you see here. At baseline, these neurons are highly active. After food presentation, there is this nice decrease in their spontaneous activity. So we went on and gavaged these mice with the MDP control. That, remember, it does not affect the not-Chiu neurons. And we could not detect any difference. However, once we gave the MDP that does activate the not-Chiu neurons, we could see that this neuropeptide would decrease the spontaneous activity of these neurons, similar to what we see when we give food. We quantified this data and we confirmed that indeed MDP would decrease the activity of these neurons. And moreover, we also saw that this decrease in activity was dependent on the not-Chiu receptor. Because when we performed the same experiment on the conditional knockout that don't have the not-Chiu receptor on the inhibitory neurons, there was no difference between gavaging with MDP control or with MDP. Lastly, we needed to demonstrate that this was indeed a direct effect, a cell autonomous effect. So in order to do that, we use the patch clamp system, in which you evaluate a single neuron. And the nice thing about this is that we were able to inject directly inside the neuron using the patch pipette our compound, either the MDP control or the MDP. And then we would record this neuron. And what we saw was this striking decrease in the firing pattern of the neurons 30 minutes after MDP infusion, a decrease much stronger than what we saw when we gave the control molecule. So we saw that MDP affects neuronal signal, that it does decrease the spontaneous activity of the inhibitory neurons of the ARC, and that it is a direct effect. So next, we decided to address which brain areas were actually involved in the neuronal nurture control of feeding and temperature. And then we took advantage one more time of the IDSO clear map system, but by this time comparing our conditional knockout mice that do not express the nod-2 receptor on inhibitory neurons against their litter-made controls on a steady state. And what we saw was that besides the arcuate nucleus, another important hypothalamic nucleus, the dorsomedial nucleus, was also differently activated in between these mouse. The dorsomedial nucleus of the hypothalamus is also important for feeding and temperature control. And as I showed for the arcuate, it also expressed nod-2 neurons in the nod-2 receptor on the inhibitory neurons. So in order to analyze if those two nuclei were indeed important for the phenotype that we had observed in our conditional knockout mice, we used stereotaxic injection of a Cre-expressing virus in a nod-2 flux mice, where we could, by injecting directly into the DMH and the arcuate nucleus in this nod-2 flux, we could remove the nod-2 exclusively from the neurons of these two areas. And as I demonstrated in the conditional knockout, the absence of this receptor exclusively on these two brain nuclei, we were able to see that they would show a weight gain, an increase in food consumption, and a decrease in their variation in body temperature, implicating these two brain nuclei as controlling this phenotype. And to demonstrate the involvement of the gut microbiota in these phenotypes, I treated these mice, the injected ones, with antibiotics for 12 weeks. And I revealed that in the absence of a microbiota, we could not detect any difference in weight gain nor in appetite. However, upon antibiotic removal, the weight difference were seen again, and also the differences in appetite, indicating that the microbiota was indeed playing a role in this phenotype. So putting all of this together, what my work showed is that once you eat, there is this increase in bacteria. And with this increase, there is this increased release of neuropeptides that somehow are able to reach the brain. And in the brain, in the hypothalamic regions, it affects the firing activity of inhibitory neurons. Once the nod-2 receptor from these neurons are activated by the muramyl dipeptide, they decrease their firing activity. And this decrease in signal is sensed by the mouse as a stop eating signal. And it also affects the body temperature. We think that this might be a way of the host to control the growth of the gut microbiota, or it might be the gut microbiota controlling its own niche by controlling how much the host eats. This work would not have been possible if it was not for the collaboration of experts from different fields. So I'd like to thank the 4 labs that I worked with and all the people that collaborated with me and all the financial agencies. I also like to thank you once again for your attention, and I'm very happy to take any questions.
Great job. That was an amazing talk. I was really interested and so you saw the difference with the older female and the younger female, and then you saw no difference in males. I'm wondering if that had anything to do with the integrity of blood-brain barrier, if you saw any differences in the immune reaction. Just curious for your comment on that.
Yeah, that's the question I wanna answer now, but we did not check the blood-brain barrier, neither the gut barrier. But it definitely could be, because there are words that describe a difference, sex difference in the permeability.
Really cool. So we have a few questions in the chat. Vivian's asking, why did the weight gain only occur after 7 months? Does the younger females and males have a high metabolic rate?
I don't think it's the metabolic rate, because we did some experiments using metabolic age and we could not detect striking differences. One thing that we are working on, it might be the hormonal effect, because it's in mice, it's around this age, 6 to 8 months, where they start to enter in the menopausal-like state. So there is this decrease, for example, in estradiol, and we know that estradiol also have a strong effect as an anorexic hormone. So we think that maybe once this hormone is not there anymore to stop us from eating, then the MDP that has a lighter effect compared to this hormone, then we can see this effect. But this is all a speculation at the moment.
Mm-hmm. Awesome. And then the other question is, how does neuropeptides reach the brain?
That's the second question I want to answer. Why only females, and how does it get to the brain? We have no idea.
And then if you challenge the begat NOD2 animals to bacteria infection, how is the immune response activation and then control the infection?
That's a great question. We never tried any infectious models, because I was actually only interested what happens on homeostasis. But there is a big difference between MDP given orally. It does not activate the immune system compared to it given IP, for example, or IV. So honestly, I have no idea. I would say that wouldn't change much, because the immune system is not affected, and the immune system will be the barrier for the infection. But it's a good experiment to try.
Anyone have any other questions? Well, this was an awesome talk. Both presenters did an excellent job. I learned so much personally, so this was really great.